Hello, welcome to Cancer Research Institute's webinar series, Cancer Immunotherapy and You. Thank you for joining us. Today's date is Tuesday, January 19th, 2016, and the title for today's webinar is Killing Cancer, Immunotherapy for Leukemia and Lymphoma. In this webinar, we will discuss the current immunotherapy treatment options for patients with leukemia and lymphoma. The Cancer Immunotherapy and You webinar series is made possible by our generous sponsors, LabAnswer and its employees, and Celldex Therapeutics and Regeneron. My name is Emily Helk, and I'm the online community manager at the Cancer Research Institute. CRI is the world's only nonprofit organization dedicated exclusively to harnessing the power of the immune system to conquer all cancers. We fund scientists around the world whose work has led to significant breakthroughs in treating cancer with immunotherapy. Over the next 30 minutes, you'll hear from an immunotherapy expert about how cancer treatment is undergoing a revolution thanks to these breakthroughs. After a 20-minute presentation from our speaker, we'll open the discussion to questions submitted by you. You can pose your question at any time throughout the presentation by typing in the Q&A box you see on your screen. Today's webinar is being recorded and will be made available on the Cancer Research Institute website and on our YouTube channel. Now, it's my pleasure to welcome today's expert speaker. Dr. Ronald Levy is a Robert K. Summey and Helen K. Summey Professor of Medicine and Director of the Lymphoma Program at Stanford University School of Medicine. For more than 25 years, his research has focused on lymphoma. He was the first to successfully treat cancer with a monoclonal antibody and went on to help develop Rituxan for the treatment of lymphoma. Dr. Levy has published over 300 articles in the fields of oncology and immunology, and we are very grateful that he could join us. Welcome, Dr. Levy.
Thank you very much. So what I wanted to do was review with you today how immunotherapy is become one of the principal ways of treating cancer, and particularly lymphoma and leukemia. So we have surgery, radiotherapy, and chemotherapy, and more and more these days what are called targeted therapies. Immunotherapies are a form of targeted therapy, but they really extend our power in addressing the immune system of the body as well as the cancer cells in the body. I guess I changed the slides here. Thank you. So there's really been a paradigm shift in our way of thinking about treating cancer. Historically, on the left here, we have the tumor cell, and we have therapies which go after the tumor cell in the body and try to distinguish it from normal cells in the body. And in the diagram here, I show an antibody molecule, but it could be drugs or chemotherapy or radiation treatments aiming at the tumor cell. But now, in addition, we have this new concept targeting the immune cells of the body. And again, I show an antibody molecule here, but it could be other forms of stimulating the immune system. And so once stimulated, the immune system can become like a drug and go after the cancer cells. So we have this dual way of approaching the cancer now, targeting both the tumor cell and targeting the immune system. Last year, on the cover of Science magazine, we had this picture and this story which called it the breakthrough of the year, Cancer Immunotherapy, T cells on the attack. And in this diagram, we see the cancer cell here on the right, and we see these molecules with elements that can recognize the cancer cell and go after it and kill it. That's an artist rendition. So we have several ways of doing this. One is through the use of antibodies, molecules that float around in the blood and are part of our immune system against bacteria and viruses. And we can make these antibodies in the laboratory now and make them in large quantities and use them like drugs. Then we have what are called chimeric antigen receptors. These are ways of programming the cells of the immune system to recognize cancer in a genetic reprogramming operation, and I'll tell you about that in detail. And then we have ways of blocking what we call the checkpoints of the immune system, the things that are keeping it from fulfilling its complete potential, ways of blocking those checkpoints. And then we have combinations of all of these strategies done together. So first, monoclonal antibodies. This is a story that began long ago, and this is a timeline in this arrow here. It really began from my point of view in 1975, when two scientists in England figured out how to make the immune system, cells of the immune system, immortal, live forever. And the products of the immune system, the antibody products, can be harvested and grown and scaled up. And if you find the right antibody that can target the cancer cell, you can make it into a drug. And we found that in 1981. And since then, several products have been approved and are now in routine use in the clinic. Rituximab for lymphoma, trastuzumab for breast cancer, and a variety of other antibodies along the way have been approved by the FDA. So this arrow goes to 2009 and obviously continues to the present time. We have new developments in the antibody field, some of which I can summarize for you here. So the products that have been approved by the FDA include rituximab for B-cell lymphoma and two other antibodies also for B-cell lymphoma. We have antibody drug conjugates, that I'll show you what that means in a minute, where we put a drug on an antibody as a piggyback to deliver it to the cancer cell, again for lymphoma. And we have new antibodies that have been engineered to recognize both the cancer cell and the immune system at the same time, and to bring the two together to help the killing function. So here's a diagram showing antibody drug conjugates. So we have antibodies floating around in the blood, looking for cancer cells, and they have ability to recognize, like a piece of Velcro on their tip, that can recognize the target on the cancer cell. And here in red shows the drug that's linked to these antibodies. And so this is a way of delivering the drug to the tumor cell using the antibody and its ability to recognize as a way of targeting the drug and keeping it away from the normal cells in the body. Occasionally, the antibodies go not to the tumor cell, but also to the normal cell. And occasionally, the drug comes off the antibody, either in the blood or in the targeted cell. And the drug can freely float around the body and hit the normal cells too. So we have both good and bad elements of this strategy. And it's working in the clinic, but it has some limitations due to the side effects from the drug that comes off. Chimeric antigen receptors are a new and very exciting way of re-engineering the immune system. What they are is cells from the body that are taken out of the blood, put in the test tube, and transduced or changed with a virus that goes inside the cells and brings a receptor from an antibody molecule into the T cells of the immune system. Then these cells are made into large quantities, each one containing this new receptor that's been put in there. And then the cells are put back in the body like a blood transfusion. And they go throughout the body looking for the cancer cells and stay in the body for a long time and increase their numbers and their patrolling power to attack the cancer cells. Here's a series, a graph showing a series, what happened to a series of patients who were treated with such chimeric receptor T-cell therapies. Every bar it represents what happened to one patient. And if the bar goes down, it means the tumor shrank. Here, 100%. So here we have a bunch of people who had a complete elimination of the tumor from their body. And here are a bunch of people who's had somewhat less than, but a really important shrinkage of their tumors. And only one person out of all of these who had the tumors growing in the body rather than shrinking. So this summarizes a really impressive result, and particularly when you know that these patients who were treated this way had been through the usual treatments and had not responded to them, and finally were treated this way and had these wonderful responses. And I should say that these are not just shrinkages of tumor, but they're actual disappearances for long periods of time. So the responses are really dramatic, not only in the amount that they shrink, but in the amount of time that the tumor is eliminated from the body. Checkpoint blockade is one of the latest elements in this story, and it's really an exciting one that has a lot of people interested. You can think of the immune system as a car that has the brakes and gas pedals. So you can push the immune system by pushing on the gas pedal, or you can relieve the brakes. And the brakes are what we call the checkpoints. So PD-1 is the name of one of these checkpoints. And we have antibodies now, antibodies that can block the checkpoint, block the brakes, take the brakes off. And so the immune system can be stimulated to go and by pushing on the accelerator or by relieving the brakes. And here is a list showing in the middle the immune system cell, the so-called T cell, and all of the brakes here on the right in colors, and all of the antibodies that have been made against the various brakes. And on the left, we have all of the accelerators. All of the targets that these antibodies here on the left can bind to and stimulate the T cells of the immune system to go faster. So this combination of taking the brakes off with antibodies over here and pushing on the gas pedal with antibodies over here can result in the T cells of the immune system being more activated and dividing and replicating and going after the cancer cells that they know how to recognize and kill. So now we have two products already approved by the FDA, one against this target called CTLA-4 and one against this target called PD-1. And these are just the first two. All the rest of them are in testing phase now in people, having worked already in animals, and are in clinical trials to see how they can also have a good effect. And eventually, many of these are going to turn into drugs that we use in the clinic. And used alone, they're going to have their independent actions, but used together, we think they're going to combine and have a really potent combinatorial effect against the cancer. So here's one of the diagrams that we use to explain all of this. Here we have the PD-L1, one of the partners that's on the cancer cell, and the PD-1, the other partner, which is on the immune cell. And the immune cell is waiting to kill this tumor cell, but is being instructed not to do that by a signal that we can call a don't kill me signal coming from the cancer cell to the immune cell. And by blocking this don't kill me signal with an antibody, which either targets this one or targets that one, either way, you can block this signal with this antibody and take the brakes off this T cell that's ready to kill the tumor cell. The poster child for this approach is Hodgkin's disease, or Hodgkin's lymphoma. Hodgkin's disease is a malignant disease of the lymphoid system that in the early days was always lethal. All patients with the disease used to die of this disease, but we have many treatments that are really effective for Hodgkin's lymphoma, and most patients today are cured with the treatments that we have. But this approach of the immune system works especially well in Hodgkin's lymphoma. Here's an example of a scan of a patient on the left, a so-called PET scan. And many things that you see here are normal: the kidneys, the heart, the brain. But these are the abnormal things here. These are the Hodgkin's lymphoma tumors growing at different places throughout the body. And just to show you how dramatic this can be, after treatment with this drug called nivolumab, which is one of these antibodies against the checkpoint of the immune system, taking the brakes off the immune system, here on the right is after the treatment, 6 weeks after the treatment. For a person who had been through all the usual treatments that stopped working and now dramatically has totally eliminated the Hodgkin's lymphoma from this person's body. So this is a particularly dramatic example, one example. But we have many examples, not only in Hodgkin's lymphoma, but in also other kinds of lymphoma and also other kinds of cancer. We have a lot of questions about this treatment. We're excited about it, but we have some questions that are listed here. For instance, how do we know who's going to respond to this and who's not? Some people respond very dramatically and some people not at all. And sometimes it has to do with how much of the target there is on the tumor cells or on the other cells around the tumor. We call them stromal cells. The more of this target there is, the more likely the person is to respond to blocking this target. But we have some people who don't have any of this target and still have responses to this treatment. So we're always looking for a better way of predicting who's the right person to use this treatment on. There are also examples of some whole kinds of tumors that respond, like lymphoma that does respond, but myeloma, another kind of tumor, does not. And we have other examples of stomach cancer responding, but colon cancer not responding. So whole categories of cancer can either respond or not, and we still have questions about why that is. Sometimes we see the tumor grow after we treat them, and it's only temporary, and then eventually it goes away. So we call this pseudo-progression. It means we think it's growing, but actually it's responding, and it's eventually going to go away. And this is a very interesting thing that when we see it, it's hard to recognize it as really on the way to a response instead of on the way to a failure. We need to understand this better and diagnose it better. So we'd like to increase the frequency that this works. We'd like everybody to respond to this. And so one way is to increase the target, PD-L1. The other way is to combine it with other treatments. So in combining these treatments, we want to make sure that they work together in a good way and not work together in a bad way. Sometimes they might interfere with each other, these combinations of different treatments, and sometimes they would work to help each other work. And we want them to work to help each other against the tumor, but not the side effects of the treatments. So we have one very recent example where we found a way of combining these treatments, where we have a small molecule diagrammed here, which is called ibrutinib, which is a drug that we use every day to treat lymphoma, certain kinds of lymphoma in the clinic. And we have the antibody that I've been talking about, the anti-PD-L1 antibody, which takes the brakes off the immune system. What we found in animal studies is that if we put the two together, we can get a really enhanced, greater effect, not only against lymphoma, but also against other kinds of cancer, like colon cancer and breast cancer. So we call this synergistic, synergy, meaning helping each other work, and together is better than the sum of each used alone. And so we're pretty excited about this. We're very excited about this, and we think it's going to be the way forward for making more people have a better result from the combined therapies using the immune system. So in a diagram shown here, this is what we want to achieve. Currently, a lot of our treatments for cancer can make people live longer. This is a curve that shows the survival of people over time who have a certain kind of cancer, just for discussion purposes. This is not real data. These are kind of a discussion slide. So we can make people live longer, but we really don't cure them with some of the current targeted therapies. That happens to most of the people who get these targeted therapies. And then we have the immune system therapies that I've been discussing today. For a certain number of people, we can have very, very long-term effects that last for a long time, maybe even cure them. We'd like to put the two together and maybe get this result up here, and that's what we're all aiming for. I really think this is going to be possible, and we're starting to see evidence of this already in our experiments that we've done in animals, and we're now testing these concepts in people in clinical trials.
Thank you so much for your presentation. You mentioned clinical trials in your last slide, so why don't we start there? We have a question from a patient. At what point should someone consider a clinical trial? Should it be upon diagnosis, in the middle of treatment, when they're in remission? When's a good time to explore that?
That's a really good question. We have a lot of treatments that really work well, and we wouldn't want to deny people those treatments that we really have confidence in and have been already approved. So we have situations where those treatments have already been used and stopped working. And most people agree that's a good time to try something new. But there are situations where we would like to try something new in the very beginning of the treatment and make the beginning treatment work even better. So we have trials of both kinds, and it depends upon which disease and which situation and which opportunities we have. I hope that answers the question.
Yeah, I think so. And who should patients be consulting with about this? Their regular medical oncologist, or should they see an immunology specialist?
Well, we have trials of various kinds of treatments, not just immune therapy treatments. So usually the oncologist or the hematologist for, in this case, for leukemia and lymphoma, or the oncologist for other kinds of cancers, knows about all the trials that are going on and can easily find them out. And they're also available on the various disease websites that people can go to and look them up. And there's a trial, a national registry of trials called clinicaltrials.gov. That's the website, clinicaltrials.gov. And that lists all the clinical trials that are going on all over the world.
So that's a resource you'd recommend for kind of first look?
Yes, that's where I go to look for trials that I don't know enough about. That's where I start.
Great. We have a really great question here. Are immunotherapies intended to replace the current standard of care or to be added on top?
We have both opportunities. It depends on the situation, on which disease and which side effects and which treatments we have available already. In some cases, our treatments are not very good at all, and we'd like to totally replace them. In other cases, we have very good treatments that we'd like to make work better, so we'd like to add to them. And we have examples of both. And it really depends on, as I said, the exact situation, the exact kind of disease, and the exact kind of treatments we have available.
So we received a number of questions about specific subtypes of leukemia and lymphoma. So could you speak a little bit to, are immunotherapy drugs effective for multiple types of cancers, or is it cancer specific or subtype specific, or how should patients kind of begin to understand that piece of it?
We have a possibility that the immunotherapies will work for all kinds of cancer, but we already have results that are very promising in some kinds, and we really want to jump on those results and extend them and confirm them. And so we're already getting a sense of where it tends to work and where it tends to work less well or even better. And this is a very fast-moving field. Even I get new information almost every day as we go to meetings and hear about new results. So this really applies eventually to all kinds of cancer. Some need it more than others, and some it works better more than others.
In your experience, we had a question about positive results, and I know this number probably varies a lot over cancer type and individual patient, but what percentage of patients using immunotherapy for leukemia and lymphoma are you seeing experience positive results?
Again, this is hard to be general about. We have various kinds of leukemia and lymphoma, and we have various kinds of treatments that work more or less well already. We have, basically in lymphoma, we have two kinds of diseases. We have the kind that grows fast and would be very dangerous if they were left untreated. And for that kind of lymphoma, we have a lot of chemotherapy and antibody treatments that work very well now, and about half the patients with that fast-growing kind of lymphoma are being cured with the current treatments. And then we have a group of those patients who are not cured, and those are the ones that we're texting immunotherapy on, and we're seeing very, very good results. Then the other kind of lymphoma grows very slowly, and sometimes we just observe and don't use any treatments right away, what we call a watch and wait approach. And there, we also have good results with immunotherapy, and it's used when the time is right, when the patient really needs to be treated with something. And we have a lot of very good treatments, but they don't cure the patient. So we're hoping to add immunotherapy to the treatments we already have and have a chance to cure people more often.
Yes, that would be huge. All right, I'm going to shift gears a little bit here to some more kind of day-to-day questions. We had some patients who were curious about access to immunotherapy, where they can be treated with it in their community, and also, the financial aspect. Does a patient have to pay to be in a clinical trial? That kind of thing.
Yeah, so access has to do with finding out where the trials are going on and contacting the people who are in charge of those trials. So clinicaltrials.gov is the site that lists the trials for every disease and then the places that they're happening and the way of contacting the people who are in charge of them. Usually they're going on in many places around the country and around the world. As far as paying for it, clinical trials are usually paid for by the sponsor of the trial, either the drug company that has the drug or the government that sponsors the clinical trials. Usually insurance will pay for treatments that are already approved by the FDA and are already proven to work. For things that are still being tested and have not yet been proven, those costs are paid for by the sponsors of the trials.
Right. I think that's important for a lot of our viewers to know. Thank you for that. And in terms of finding a clinical trial, are you seeing them mostly at only the major cancer centers, or are they in the more community setting as well?
They're in both. The major cancer centers all have clinical trials. That's their purpose. And they attract sponsors with new agents to be tested. But the sponsors are also enlisting consortia of private practitioners in the community away from the major centers. We want to know if things work for everyone, not just for the people who can travel or make it to one of the major centers. So eventually these trials are done more broadly, involving people everywhere in the private practice community setting.
Right. So patients really can access this in their own community and shouldn't feel like they'll have to travel to a major city or anything like that to participate.
Again, it depends on the details of which trial and which treatment. Some of them are only in one or two places around the world, and some of them are being studied in thousands of places around the world.
Okay. So I just have one more question for you, and what are you most excited about in the coming down the pipe in the next year or 2 years? What's the science that's just really thrilling to you?
Well, I ended my presentation with the thing I'm most excited about. It's the putting together of these various elements that are working and giving results already, but putting them together in ways that are going to make them work even better. The example I gave of the combining ibrutinib with the PD-1 antibody blocker is something that we developed results on in our own research, and we're quite excited about it and think that it's going to work in the clinic, and it's being tested now, and we're part of that testing process.
Great. Well, thank you so much, Dr. Levy, for your participation and for your wonderful presentation and for being so generous with your time as to share all of this information with our audience. I really appreciate it.
Thank you for having me.
Be sure to visit theanswertocancer.org, our portal for patients and caregivers. You can learn about immunotherapy and clinical trials for different types of cancer and hear directly from other patients who've been treated with immunotherapy. Thank you once again to our generous sponsors, LabAnswer and its employees, and Celldex Therapeutics and Regeneron. Thank you, Dr. Levy, and all of those who joined us for today's presentation. If you'd like to see more expert presentations on how cancer immunotherapy is transforming treatment, visit www.cancerresearch.org/webinars. These slides will be available to all participants after a short survey you'll be directed to at the end of this webinar. We hope you'll take a moment to provide us feedback. Thank you. Have a good day.